Dan Kastner, and I had the auspicious role of moderating the sessions today, the medical and scientific sessions, while you all were in your own sessions, which I know were also very good and very interesting as well. But I thought that what I would do over the course of the next 5 hours is to break things down, I'm just kidding, into 5 parts. First of all, I'd like to say a few words about what the Dada 2 Foundation has done and its impact in terms of the research and the progress that's being made in the field of Dada 2. The second thing that I would like to do is the second, third, and fourth things that I would like to do is to talk about 3 general areas of the abstracts or the talks that we had over the course of the day today. And so altogether there were, I think, 26 different talks that we had today. And they could be broken down perhaps into 3 different groups. One of them had to do with diagnosis of Dada 2 and the emerging spectrum of different phenotypes, different symptoms and signs of Dada 2, different manifestations of Dada 2 that we're learning about. That was one of the areas, and there were 12 talks altogether that had to do with that. Then there were 9 talks that had to do with emerging things with regards to the mechanism of Dada 2 in terms of the different pathways that may be involved that make people who have Dada 2 have different clinical manifestations. And then the last 5 talks, so 12 plus 9 is 21, plus 5 is 26, so there were 26. The last 5 of them had to do with various aspects of treatment of Dada 2. And then finally, at the end, I thought that since we're all here, I'll try at least to field any questions that you may have that maybe I won't cover just because of the fact that I'm giving sort of a general overview of what happened. So in any case, to start out with my first topic, which is the impact of the Dada 2 Foundation in these different areas of research that I've just mentioned. I must say that, you know, having been here at the first International Congress on Dada 2 2 years ago, it's breathtaking to see the progress that's being made, and it is just so palpable, so tangible, the progress that's being made as a result of having these meetings and the things that the Dada 2 Foundation is doing. And for example, in the area of understanding the spectrum of Dada 2 and understanding how to diagnose Dada 2, certainly the fact that we have now a community of physicians and scientists who get together and compare notes about Dada 2 and how to make the diagnosis, and comparing notes with regards to what patients with Dada 2 present with in terms of their clinical manifestations. That was really very, very much potentiated by the meeting that we had 2 years ago. And I see that just in terms of the interactions that people have that I'm aware of between these meetings, emails that go back and forth between people, collaborations that are developing, and various talks that were presented today that represented an outgrowth of things that were talked about at the last meeting. So that's one area where the foundation certainly has had a major impact. Secondly, in the area of pathophysiology and mechanism of disease, that also is an area where the foundation has had a major impact, and certainly one of the talks that we had at the last meeting from investigators in Norway having to do with Dada 2 perhaps having a role as a lysosomal storage disease. We've seen that concept really blossom over the course of the last couple of years, and we've learned a lot more about that. And I see that as really having been a consequence of what was presented a couple of years ago. And then finally, the area of treatment. I think that there's no question that the interest in bone marrow transplant, at least for some patients with certain manifestations of DADA2, and the increasing interest in developing gene therapy for DADA2, that those have very much been potentiated by the things that the foundation is doing, encouraging people to develop research programs in those areas. So then maybe to talk about the three topics, the three general areas that I mentioned at the outset. The first of them being diagnosis of DADA2 and the clinical manifestations of DADA2. There was a wonderful talk that we started out with in the morning by Taryn Youngstein from the UK, just talking about, it was, again, very much catalyzed by the foundation. It was a talk having to do with a survey that was done, a Delphi survey that was done among investigators, among clinicians who see patients with DADA2, and if they have agreement with regard to how the disease is diagnosed and how the disease is treated under certain conditions. And certainly, although there's a lot of discussion and debate with regard to certain details of that, I think that in general, there was consensus that was reported with regard to the fact that both genetic testing and enzyme assay levels have a place in terms of making the diagnosis. And right now, neither one of them alone is probably the be-all and end-all with regard to making the diagnosis. And the reason for that is that we have some patients where when we do genetic testing, we'll find a single mutation, and as you know, it's a recessively inherited disease, so we would expect usually to find 2 mutations, and sometimes we only find a single mutation, and it's hard to find sometimes the second mutation, even if it's there. And so that means that there is at least some work to be done still in terms of the genetic testing approach for DADA2. On the other hand, with regard to the functional assays, you might say, well, then let's just do the functional assays. And the problem with that is simply, and we had a discussion about this this morning, that although the enzymatic activity does give us a readout in terms of how much ADA2 is there in the blood, we don't know necessarily that all of the clinical manifestations of DADA2 are derived from that enzymatic activity. There may be other activities of the DADA2 protein that we don't yet know about, or that we're just beginning to imagine about, that may be very important in terms of whether or not one has DADA2. And so, again, that isn't the be-all and end-all either. We also had a discussion about whether we should do, at this point, newborn screening for DADA2, and whether we should do some sort of sequencing where, you know, we could sequence a whole population of people looking to see if they have mutations in the gene. And it was felt that although we might learn something at a research basis from doing those kinds of things, that right now, until we have a little bit better concept of exactly how the disease works, that it's a little bit too early to do those things. But certainly we're thinking about how to do them, and it might be that as a result of these discussions that someone would actually then do a research study looking at 1,000 individuals, you know, who don't have any symptoms, to see if they have mutations in the DADA2, in the ADA2 gene, and if so, if we went back to them, that maybe they would have some problems that they haven't sought medical attention about. And so these are the kinds of things that these kinds of discussions get us thinking about. It also gets us thinking about, well, you know, if somebody in the genetic testing has a single mutation, and really that's all we can find is a single mutation, then, you know, could there be, under some circumstances, could there be some sort of clinical manifestations that would go along with that? And certainly we heard, at least in one of the talks, about a possible mechanism where maybe that could happen under some circumstances. We don't think that it happens under a lot of circumstances, but maybe in certain circumstances that would happen. And certainly understanding that a little bit better is going to be very important, and certainly for those of you who are family members of DADA2 patients where you may yourself have a single mutation in the gene. That's certainly, I know, a major question that we've been dealing with ever since we first discovered DADA2 back in 2013 or whatever it was, and talking to parents and grandparents of children who have DADA2 and wondering whether or not they're at any increased risk for stroke or any other clinical manifestation. It's something that we have to think about, but we don't have at this point because we don't quite understand the disease well enough. We can't say exactly what would be the recommendation, but again, that's the basis for more research in the area. We've seen that, you know, whereas DADA2 started out as a couple of sort of primary clinical phenotypes, if you will, clinical manifestations, those being fever and stroke, as was reported by the NIH series, and polyarteritis nodosa, vasculitis, as described in the Israeli series. Those were the two things that we started out with, and we now know that there are a number of other things, and we heard about them this morning, a number of other clinical manifestations of DADA2. So just thinking about the polyarteritis nodosa, we heard a talk, it actually wasn't in the morning, but in the afternoon, about how adults with polyarteritis nodosa, sometimes adults that may be in their 20s or even 30s who are diagnosed with polyarteritis nodosa, don't have any family history of it, can turn out to have DADA2 on genetic testing. And so it's just an example of how once you know how to look for something, you actually can find it then in people where, you know, before that, you wouldn't have dreamt that something like that was going on. Also, we certainly now know that there are other phenotypes that are associated with DADA2. We know that there can be problems with the bone marrow in terms of either the differentiation of red blood cells or sometimes subsets of white blood cells that can sometimes be refractory to some of the anti-TNF treatments that usually work well for stroke and polyarteritis nodosa. We also know that there can be immunodeficiency phenotypes that can be associated sometimes with DADA2, and that there can be, not commonly, but there can even be lymphoproliferative phenotypes that are associated with DADA2. And certainly one of the to-do items that came out of the discussions this morning was the idea that we really need to have a worldwide registry of DADA2 where it's for people from all countries of the world and where it's all the different manifestations that we know of, so that it's not that you have people that are studying stroke in the United States, you know, in one cohort, and the people who are studying polyarteritis nodosa in the Middle East in another cohort. It has to be all of the different manifestations that we're aware of in all of the countries of the world, so that we can get a better idea of how all of these things relate to one another, and to what extent could, for example, one phenotype sometimes progress into a different phenotype. In other words, could someone start out with polyarteritis nodosa and then later on have a problem with some other aspect of DADA2 that I've mentioned in the list of possibilities for DADA2. So that was the first set of abstracts, or talks. The second set, which was 9 talks, had to do with the biology and pathophysiology of DADA2. And let me just remind you that the thoughts that we had, at least initially, with regard to the mechanism by which mutations in the ADA2 gene might cause illness in patients, that there were a couple of sort of signal ideas there. One of them was that ADA2 might be a growth factor for endothelial cells. And you all may remember that there were the studies in which the ADA2 gene was inhibited in the embryos of developing zebrafish, and the zebrafish developed strokes. And so that was fairly persuasive evidence that perhaps ADA2 is a growth factor for the cells that line the blood vessels, and that if you don't have enough ADA2, then that can lead to problems with blood vessel development. And then there were also the studies looking at the differentiation of certain subsets of white blood cells. And the idea from that was that you can have skewing. There are subsets of white blood cells that are pro-inflammatory and that are anti-inflammatory, and that when you have a deficiency of ADA2, you get a skewing towards more of the pro-inflammatory types of white blood cells. Those ideas still persist. It's not that we're in any way saying that those things are not true, but there are now new concepts that are coming about with regard to how perhaps mutations in the ADA2 gene could lead to illness. And so one of the concepts that really has taken off is the idea that, in fact, in some way, that ADA2 deficiency might lead to an excess activity of something called type 1 interferon. And type 1 interferon is a substance that's made by the body, oftentimes as a defense against viruses, actually. And type 1 interferon can cause a number of inflammatory manifestations, and there might be specific treatments even that target type 1 interferon that we could think of in treating patients with DADA2 who don't respond to other therapies such as anti-TNF treatment. So that's a concept that certainly is coming about. And one of the ideas that has taken off is the idea that, in fact, ADA2 may be an enzyme that lives in a certain part of the cell called the lysosome, and that it's involved in degrading DNA, and that if you have a deficiency of ADA2, then you don't degrade the DNA right, and so you have this free DNA in the cytoplasm of the cell. And free DNA can sometimes be recognized as if it's a virus, and that stimulates type 1 interferon, and that leads to this interferon kind of process. So that's one of the things that certainly we were talking about a lot. We were also talking about the role of adenosine itself in terms of stimulating some of the pathology of ADA2. And then there was an interesting idea, netosis. So there's a certain kind of white blood cells called neutrophils that sometimes when they're stimulated, they explode and they extrude DNA into the area around where that white blood cell was. And actually that DNA, for those of you who have ever worked with DNA is really sticky. And so the idea is that it would be sort of like a sticky medium that would capture bacteria and prevent them then from infecting cells. So there's a purpose for having this process of extrusion of DNA. It's called netosis, and actually there have been some studies now looking at netosis in patients with DADA2 and seeing differences in netosis in DADA2 patients relative to healthy control individuals. There was also a very good discussion about what cell types actually are the culprits with regard to some of the manifestations of DADA2, and we're still, even at this point, having discussions about whether the endothelium, the cells that line the blood vessels, in fact, express significant amounts of ADA2 or not. And if they do, then that may have an impact in terms of why you get blood vessel disease, or it may be, as I said at the outset, that really ADA2 is something that's made by white blood cells and that it's a growth factor for the cells that line the blood vessels. It has some import in terms of then how you would think of treating the disease eventually. So although this may sound like it's a purely academic discussion. In fact, it does have some important implications in terms of treatment. And certainly this whole idea of the interferon involvement with DADA2, there is a class of drugs that's called the JAK inhibitors, which are actually used for the treatment of certain interferonopathies, as they are called, in other clinical settings. There are certain other diseases that are caused by excessive amounts of this type 1 interferon, and at least to some extent they do respond to these inhibitors of something called JAK kinases. So these are certainly advances in terms of the biology that could lead eventually then to advances in terms of treatment. So then leading there into the third area, that being the last 5 talks that had to do, at least to some extent, with treatment. What can I say about that? Well, I guess there were 3 general areas of treatment that were discussed. One of them was the use of TNF inhibitors. The second was the use of bone marrow transplantation. And the third was the possibility of gene therapy. So first of all, with regard to the TNF inhibitors, I think that both from the survey that was done in the very first talk and in discussions that we had in the afternoon in the treatment session, there's fairly good consensus, even if the overall data is perhaps not as plentiful as we would like it to be, but there is consensus that TNF inhibitors are effective in preventing stroke and in treating the inflammatory manifestations of DADA2, such as polyarteritis nodosa. But certainly another of the to-do items that we took away from the meeting this afternoon was the idea that we really need to aggregate all of the data that we have from all over the world with regard to responsiveness to TNF inhibitors, so that that data can be given to the regulatory agencies and to third-party payers, so that it will be easier for people, for patients and their families, to get TNF inhibitors for treatment for DADA2, because there was a lot of discussion about the fact that it's hard still, even though experts on DADA2 may agree that this is the right thing to do for that manifestation of the disease, it's still hard to get funding to get those medications for patients. And there was a very thorough discussion about what TNF inhibitors can do and what they can't do in terms of treatment for DADA2, but certainly the things that are inflammatory mediated are, we think, very susceptible to treatment with TNF inhibitors. There are, though, patients that have problems with, for example, bone marrow manifestations of DADA2, the diamond black fan anemia-like presentation, the pure red cell aplasia, the neutropenia manifestations of DADA2 that don't seem to be all that responsive to TNF inhibitors, where in fact bone marrow transplantation may be the proper approach for those patients. And so we heard about actually now 23 patients from around the world who have been treated with bone marrow transplantation, and luckily, fortunately, all 23 of them are doing okay, although we did hear about various problems in terms of doing the bone marrow transplant. What is the right regimen for preparing the patient for a bone marrow transplant? Who should they get a bone marrow transplant from? Should it ever be from someone who's a carrier for DADA2? If we think that there could be some manifestations of the disease associated with being a carrier, maybe then we don't want to encourage carriers to be the donors in those cases. What should be the drugs that would be given to patients before the bone marrow transplant is given to maximize the chance that the transplant will take on the first round without having to do a second transplant? These are all things that were discussed. Certainly at this point, it's a subset of patients where we would think that bone marrow transplant is the treatment of choice, but it's definitely out there and is a treatment for certain of the patients. And then finally, the possibility of gene therapy. And we heard actually 2 talks on gene therapy this afternoon, one of them from Milan, Italy, and the other from London, both of them talking about sort of the building the foundations for being able to do bone marrow transplant. There's a lot of work that has to be done at first identifying, well, what cell type would one do gene therapy? If I said bone marrow transplant, I meant gene therapy from Milan and London. And so one has to think about what is the cell type that one would use, developing models in which you can prove that if you introduce your gene therapy vector into that cell type, that you can correct problems that are associated with the deficiency of ADA2, developing the right kinds of vectors. It's still a ways off in terms of having gene therapy for DADA2, but on the other hand, certainly in the discussion we had, and I did pose the question to the presenters from Milan and London as to exactly when did they think that we might have a protocol available for that, and certainly nobody has a crystal ball, so no one can say for sure. But I think that there's a consensus that the time that it would take to develop a gene therapy approach for DADA2 would be a lot less than the time that it took to develop a gene therapy for ADA1, or just adenosine deaminase, which was one of the first diseases to be approached by gene therapy. A lot of the advances that were made in developing those gene therapy approaches now can be capitalized upon for developing gene therapy for DADA2. So in any case, I mean, those were, I think, some of the high points of what we talked about today. I'm sure that there are probably topics that I didn't mention in my summary, but on the other hand, even though you know, it's funny, when you stand up here and you're talking to people, it seems like it's just seconds going by. But looking at my watch, I guess it's about half an hour that has gone by or something like that. It seems like it was just a moment ago that I started talking. But if you all have questions, I can try to answer them. Or perhaps if there are some of the presenters in the back of the room, I may have to defer to them in terms of answering some of your questions. But please, if you do have questions, we have microphones here. The rule with regard to the microphones, and we had a big deal about the microphones earlier today, so only 3 microphones can be on at the same time, or something terrible will happen in the room. I don't know what it is. We were just warned, do not have 3 microphones on at the same time. Yes, please turn your microphone off if you're not going to speak. Yes, but if you do want to ask a question, you have a microphone in front of you, just push the button, raise your hand and you know, push the button. Oh, come on, you can ask the question. Don't be shy. You don't have a question. Well, okay. I thought you looked like you were about to ask a question. Oh, he's going to ask the question. All right, well, would someone like to ask a question? Anybody, ask a question, please. Okay, we have a taker here.
You talked earlier about gene therapy, and I just want to know how far along are we in terms of gene therapy? Like, what stage are we at?
Yeah, well, so we're not at the point where gene therapy would be given to any human beings. At this point, we're at the stage of developing the gene therapy vectors, developing cell lines where we can test these gene therapy vectors, where we're looking at assays for how we would know that it works, those kinds of things. And all of that stuff has to be done before it would be ethical to do gene therapy on a patient. And so we're still in the foundation building stages. What we talked about was that maybe somewhere in the range of in the next 5 to 10 years, something like that, there could be a trial of gene therapy. But we would have to think about who would be the right patients to try gene therapy in. It may be that patients that are on a TNF inhibitor and doing totally fine on a TNF inhibitor, you wouldn't want to immediately take the chance of doing gene therapy on those individuals. So we'll have to come up with some criteria, and certainly we'll want to engage, it's an important thing to engage the community in terms of who would be the right first candidates anyway for doing it. But that was, your question was exactly the question that I asked our two presenters this afternoon is, well, when might it be? And the initial answer was, well, that's the $64,000 question. There was a TV show long before your time called the $64,000 question. I was a kid when, which was a long time ago, and that was a very popular TV show. But you know, this is the $64,000 question as to when it would be available. So, do you have a question?
How long will I be able, or how long will I have to take the medication of Humira?
Oh, my goodness. Well, now that's a good question. Now you have to turn off the microphone. You see, we have to follow the microphone rules here. The answer to that question is that although I can't speak to each person's individual medical history, I would say that in general, we think that for patients that have two mutations in the ADA2 gene and who have an increased risk of either polyarteritis nodosa or stroke. And so we could talk one-on-one about, you know, your individual situation. But for people that are on TNF inhibitors for that reason, that probably we would want to treat, at least for now, indefinitely with a TNF inhibitor. It might not be Humira, it might be a different one instead, but we do know that if we stop a TNF inhibitor for someone who is at risk for one of those things, that then the risk comes back, that it's not as if one is cured by taking Humira for a certain period of time. That's part of the reason why we're talking about gene therapy, is that gene therapy eventually would be curative. And so, you know, that is obviously the dream in all of this, is to have something that where one could just do it, you know, once, or it would not be, you know, like in a half an hour or whatever, but, you know, over a period of time, that then would fix things for good. But right now, we're not there yet. Do you have a question? No? No question. Okay. Yes, please.
And what kind of TNF inhibitor works best? Because you heard stories about Enbrel doesn't work, and you heard stories about Humira doesn't work. What TNF inhibitor does mostly work?
Well, that's also a good question. Maybe not the $64,000 question, but $32,000 maybe. You know, if you think about the potency of different TNF inhibitors, etanercept, Enbrel, is of the different possibilities that are available, it's probably the one that is the, I won't say weakest exactly, but it's the one that probably has the lowest level of efficacy. Now, that being said, it can still be quite effective in a lot of patients, and a lot of physicians would start with etanercept for several reasons. One is that there's a lot of experience with using etanercept in children, in particular, with particular rheumatic diseases, and so we know a lot about how to give it. It's also because of the fact that it's perhaps not quite as potent as some of the others, then there's less chance of side effects, you know, with regard to infection risk and that sort of thing. And so that's another reason why one might start out with etanercept. But we do, if a patient seems not to be responding after we increase the dose of etanercept to the maximum that's allowable, then we would switch to one of the other TNF inhibitors. And the other TNF inhibitors are all antibodies against TNF, rather than being Etanercept is what's called a fusion protein, a soluble receptor fusion protein. So it's a slightly different mechanism by which it works. The antibodies are, in general, more potent and last longer. So the half-life of etanercept is around 5 days. That means that if you get a dose of it, that after 5 days, the blood levels are half of what they were, when you start out. Whereas for the antibodies, the half-life is about 28 days, something like that. Varies a little bit from one to the next. So they last a little bit longer, so you can then give them less frequently than etanercept. And in general, they are more potent than etanercept, but that comes, all of these things, it's a trade-off. On the one hand, you get more potency. On the other hand, there is more of a chance of side effects.
Thank you.
Yes. Yes. Jake.
What do you believe to be the most profound discovery or milestone, in your opinion, so far?
Oh, my goodness. Well, that's like asking me to choose between my children, And, I love them both. I have 2 sons. I love them both. And, of course, we have many more children, in the sense of the discoveries over the last few years. And so I certainly don't want to, by choosing one of them, then to imply that another one isn't really interesting. I would say that, all of the things that I mentioned, there was a lot more that was discussed today. I gave you half an hour of what was, 10 hours worth of presentations and discussions. So I already have, to some extent, chosen amongst the children. It's just that I haven't mentioned, the other ones, who are also very nice and above average, and, Other questions? Yes?
Dr. Kastner, I know you said that you don't know how long it'll take, for this clinical trial, but if you had to make an educated guess to when we would actually do a clinical trial for the gene therapy, what would you guess as far as a timeline of how long it would take?
Yeah, well, so it's 2 other groups. It's not my research group, so it's not like I can say because I have, inside information or whatever. And my group doesn't, we're not a gene therapy specialty group. So 8 years? Okay, you heard it from the good Dr. Brogan. 8 years it is. And, I have, and I won't get into what they are, but I've had a number of political bets with my 2 sons, with regard to what's going on. And I bet a dozen donuts on various things. So, we could have a donut bet, with regard to the 8-year prediction. And just to be on the right side of things, I'll bet Dr. Brogan that it'll be less than 8 years, and then he can take the 8-year time point, and then maybe somebody else will take the more than 8 years, maybe if there's a pessimist in here. I'm usually an optimist about these kinds of things, though. Yes?
So when gene therapy,
When?
it becomes available,
That's the optimist.
absolutely. Do you know what that process is going to look like? Like, we know what a bone marrow transplant entails. Do we know what gene therapy might entail, the process of going through that?
Yeah, well, that's a good question. And so it probably would involve some aspect of what one does with gene therapy is that you take cells from the person that's going to undergo the gene therapy. You have to take the cells out of the person and then introduce the gene therapy vector into the cells and then grow the cells in tissue culture for a period of time. And then you have to give the person some kind of medication that will, at least to some extent, get rid of some of their own bone marrow cells so that the altered cells can go into that person, go back into that person. It's from the same person. And that they'll be able to grow and to replace the bone marrow cells that lack the ADA2 gene. So that's the process, and so to some extent it would look not completely like, but a little bit like bone marrow transplant in terms of some of its aspects anyway. And maybe Dr. Brogan would like to add something or correct me on any of the details, or you would.
I think that's a good description. We don't know for sure. The hardest question is, which of you patients will want to have that procedure first. We really need your help. It's a two-way conversation. We have an idea. We think it will be the sickest patients who don't have any prospect, who would ordinarily have a bone marrow transplant, but there's no donor. That might be a logical choice. But that's what us doctors think. You patients might have a completely different view, and you'll tell us.
Okay, all right. So other questions that people may have? Yes.
Do you think data 2 will be more common than your first thought?
Well, yes, data 2 is certainly more common than what we first thought. I mean, when we first happened upon it, we thought that it was going to be a pretty rare disease, and now it's turning out that, there are. We heard that so far from Dr. Chambers that I think he said there are 321 patients that he's aware of that have DADA2, and I'm sure there are many more. What happens is that because we have a way of looking for it, once we know what the gene is and there's a way of doing genetic testing, then it's possible to say that people that you never would have thought necessarily that they had a genetic disease, you can do the testing and find out that they have DADA2. And that has happened with other genetic diseases too. My lab has worked on quite a few and discovered a number of them, genetic diseases over the course of the last 20 years. But I think that there was general agreement today that DADA2 is on the 99th percentile end of things in terms of there being more patients than what we had thought of, and that there are more different manifestations of it than we ever would have expected, and that it's really turning into something a lot bigger than for a lot of the genetic diseases that we've found, where there may still, even a few years after discovering them, only be a few patients that have them. And even though for those diseases too, there can be testing for that disease, amongst patients that are suspected of having it, but it doesn't turn out that, there are so many as there are with DADA2. So it is turning out to be a lot more than what we had expected. And there are still the questions, oh my goodness, the goblins are here, the skeletons are here. So in any case, we still don't have a real good sense of how many people with common problems might have maybe not mutations in the DADA2, in the ADA2 gene, but some kind of a variant in the ADA2 gene that maybe leads to a little bit less in the way of levels of ADA2, but not zero amount, but less in the levels, and maybe that in some people would lead to increased risk of cardiovascular disease or stroke or something like that. We don't know. We don't know. And those are also important questions that need to be addressed in the next few years. Other questions? Yes, please.
Hello, doctor.
Yes.
My question is regarding the genetic therapy. What are now, at the date, the barriers regarding knowledge to implement the therapy in a patient? I know that there might be a money problem or something like, to develop the therapy or the investigation, but what are now the things that we need to know?
Well, I would say that at this point that it's not so much a matter that we don't have the general outlines of how to do it, but it's just that for each particular disease where one thinks about doing gene therapy, one has to go through sort of a systematic approach of figuring out what kind of cell it is that you're going to put the vector in, figuring out if there are any side effects that might develop from it, and if so, try to work out a way of minimizing those side effects, figuring out how you would treat the patient beforehand to make room in their bone marrow for the altered cells, those kinds of things, and each of those things requires some time. I mean, it's not as if we can just snap our fingers and that we would know it. So a lot of it depends on having dedicated people like the teams in London and Milan just to sort of work very systematically and diligently to work through those questions so that we can get to the point where we know all the things that we need to know. A long time ago, when I was just starting my research lab, we were working on a very difficult problem. We were trying to find the gene for a disease called familial Mediterranean fever, which was the first genetic disease that we worked on. And at that time, the Genome Project hadn't really taken off, and we didn't have maps of the human genome. We had to generate them ourselves. And there were just lots and lots of things. It was much more cumbersome to do the kinds of genetic studies then than what it is now. And in fact, if you look at the time that it took from when we first started working on the project, which was the summer of 1989, and we finished in the summer of 1997, so that was 8 years. And it was a lot of work, and sometimes it was kind of discouraging. We'd hit up against a wall or whatever. But the thing that kept me going on it, and this is what I used to tell the people in my lab, and I'll say this with regard to coming up with gene therapy for DADA2, is that, there was nothing conceptual that would make it so that you couldn't find the gene for FMF. It was just it was a big job, but there was nothing theoretically that would keep you from doing it. And so I always said that, well, you just have to turn the crank and eventually the wieners will come out. And that's what I think about this kind of thing, is that it just takes diligence and perseverance and some degree of confidence, but I think that it'll work. All right.
Can I add something, please?
Yes, please. Yeah.
I just wanted, on behalf of the patients and families, to thank you and all of the doctors and scientists who are here. We really cannot thank you enough for the effort you're putting into this, and we send you lots of hope and thanks, and we will do whatever we can to help the process. So thank you.
Well, just by you guys being here is thanks enough, believe me. It really is so gratifying to have you all here and to have a community that's so supportive of this work. And I think that I speak for all of my colleagues, healthcare professionals and scientists and laboratory investigators, in saying that we really, really appreciate having you here. Thank you.